Hello and welcome to Biofilm News Podcast, the show that brings you to the forefront of biomedical research, biotech, pharma, and healthcare fields, and the professionals behind it. I'm your host, Pavel Ryzhov. First, I guess my questions to you in regards to your work have to do with obviously your focus, which is studying the caspases in regards to the apoptosis. So if you don't mind getting us a little bit of a bird's eye perspective on the type of research that you've been involved in over the years.
Yes. So as a graduate student, I worked on a protein that was a protease inhibitor. And so at that point, early on, I became interested in proteases and their regulation. And that interest has stayed with me all the way through to today. And the reason I work on caspases is not because they're involved in cell death. I work on them because they're proteases. And I really enjoy studying the regulation of proteolytic activity in the way that proteases regulate biological mechanisms. Mm-hmm. And as we know, the caspases regulate a couple of interesting biological mechanisms, as you know, because you studied this yourself. They regulate two types of regulated cell death, one called apoptosis, another one called pyroptosis. And so therefore, they're sort of, but my interest has always been as a protein chemist, a protein engineer, rather than as a biologist, I have to say. Mm-hmm. And it's just by fluke, I've ended up in the area of regulated cell death mechanisms. And actually, I think that the early work I did in that with my collaborators, who are more biologists than I am, kind of set the scene for the fact that I'm now known to be a caspase guy. I reject that. I say I'm a protease guy, and the caspases are just one type of protease. I'm interested in all types of proteases. So my interest from the kind of the high view is in the regulation of biological events by proteolysis. Mm-hmm. Just like some people are interested in the regulation of biological events by phosphorylation or by sort of ubiquination, for example, I'm interested in regulation by proteolysis. And so have been for a long time, since I was a graduate student. And I kind of kept that interest, but shifted fields because I become interested in different families of proteases at different times.
Yeah. I have to ask, obviously, the question that I've seen in your slides and your presentation, that famous New York map of proteases. How did this come about?
So that came about because I saw something that I think Tony Hunter and his colleagues have put together called the kinome, in which they had this kind of branching network of relatedness of different protein kinases. And I thought I'd try to do something like that for proteases, and, to show the relatedness and the origin and the different groups of proteases. Mm-hmm. I tried to put it together, and it was very difficult, actually. So I asked Tony Hunter, I said, how did you do that? What kind of program did you use? He said he didn't use a program. He just had a couple of graduate students working hard in PowerPoint. And so I decided I wasn't going to dedicate a couple of graduate students to do it. So what I did is I took an existing map, which was a New York subway map, and superimposed on that the branch lines and so on, the proteases, and to show the relatedness. And so the relatedness is described very well by the New York subway map.
Yeah.
So it was just a way of kind of having people remember the different families of proteases and the kind of landscape that they occupy in the proteome.
Has this particular map and specific picture been actually published somewhere, or this is just like
Oh, it's published. Yeah, it was published in a review we put together a couple of years ago.
Oh, that's so cool. So when you say proteases, and what do you think is the most exciting part about studying proteases versus so many other different enzymes? What interests you in that?
So proteases are very easy to target. They have a specific activity that can be examined biochemically and biologically and chemically. They're really good targets for therapeutic intervention. And we know, for example, there have been some huge successes along the way, massive failures as well, but huge successes like HIV protease, hepatitis C protease inhibitors. So hepatitis C is now a disease of the past. It's been controlled by protease inhibitors. And we know that some of the sort of drugs that are used to regulate HIV are protease inhibitors as well. Mm-hmm. And the blood coagulation network. The blood coagulation network is a kind of hodgepodge of proteolytic events. And that can be controlled by protease inhibitors as well. Mm-hmm. So proteases are really good targets for a therapeutic intervention. That's not the only reason to be interested in proteases. Another reason to be interested in them is that they're really interesting. I have to tell you, but my interest in science is really discovering new mechanisms, and the mechanisms that regulate their activation and influence their ability to conduct their biology just fascinates me. And I study that because I can.
Mm-hmm.
Scientists waffle a lot about why they're interested in certain things. And what they often don't say is they do it because they're good at it, because they can do it. And this is, I've become good at it and I can do it. And so that's myself and my lab members over the years have developed, the ability to understand proteolytic events. And so we do it because they're there, as well as because they're good targets for therapy. Mm-hmm. So it's a lot of complex interactions there.
So do you see that the work that you and your colleagues have done over the years have really paved the way for and sort of laid the foundation for development of novel therapeutics targeting proteases? Is that kind of the major achievement that you see for yourself?
Yeah. So we don't tend to be a very translational lab. We're a sort of basic science lab. But in understanding the basic science, other people take forward our discoveries and can translate them. And I give you an example that you may be aware of, are IAP antagonists. Mm-hmm. So IAP antagonists are proteins, they're natural proteins that are able to reverse the inhibition of caspases by caspase inhibitors. So we actually identified some of the first IAP that inhibited caspases and proposed the use of that understanding to engineer small molecules that would ablate the interaction and therefore reactivate caspases in cancer cells. Because the inhibition of cancer, as we know, the inhibition of apoptotic cell death is one of the hallmarks of cancer. Mm-hmm. And we thought if we could activate apoptosis and programmed cell death, that form of programmed cell death in cancer cells, we might be able to derive some therapeutic benefit. And so that was later developed by a couple of big companies as to what is known today as IAP antagonists, and sometimes called SMAC mimetics. Mm-hmm. So that's one of the contributions that, understanding the basic interaction of proteins with each other leads towards therapies. Mm-hmm. So actually, that's not even inhibiting a protease, that's activating a protease. So again, understanding the natural biological regulation leads towards ways to influence enzymes for therapeutic benefits. So that's one of the examples I can give. Mm-hmm. In terms of inhibitors, protease inhibitors, I don't think we've been able to, none of our work so far has led towards the development of novel protease inhibitors.
Mm-hmm. So looking back at all the way from doing your work as a graduate student and now, what are the most profound examples of experimental challenges that you have faced over the years, and how did that sort of evolve over time from where you started to where you are now?
So what I found is CRISPR came too late. I would have liked CRISPR to have been developed 20 years ago. Because what we were doing is, what we're interested in, understanding fundamental mechanisms, and then in a very much in vitro setting, in a test tube. So we can reconstitute purified components and find out how they work. And that's great, but actually demonstrating that's how they work in vivo or in whole cells even is a very different question. And so the way that we proposed to do that was to understand the fundamental mechanisms and then go into cells and even organisms and reconstitute particular proteins with mutants of those proteins, and then test whether the hypothesis we came up with for purified components would actually result in understanding and validation in a biological system. And so the way to do that, we proposed the way to do that would be to develop a mechanism where you could mutate specific amino acids in a protein and in an actual cell. But of course, we didn't invent that. We just proposed that would be the way to do it, just as some kind of thought for the future. This was a long time ago. And then CRISPR was invented, and like 20 years after we were thinking that would be really useful for us. And so we jumped on the CRISPR bandwagon with everyone else, of course. Mm-hmm. And that is now proving to be a very interesting way of validating or actually testing the hypothesis you come up with in vitro. So I say the biggest challenge has been to translate your understanding of biological mechanisms in a test tube into a cell and an organism.
So would you say that because of the fact that you did not have techniques such as CRISPR early on, this created maybe unnecessary ambiguity in the types of discoveries that were made early on?
Pavel, that's a very interesting question. I'd rather dodge it, but I won't dodge it because I think that's a very good point. I think that probably is one of the reasons is that it did create a lot of ambiguity, because you were stuck with these proposals that you developed from very, very purified in vitro systems. And you proposed that that's the way things would happen in vivo, but it was almost impossible to test it. So I'm sure that led to a lot of ambiguity.
Please subscribe to this channel to stay up to date with latest news and with conversations with professionals from the industry.